Alright team, here we are for concept 13.3. It says meiosis reduces the number of chromosome sets from diploid to haploid. I think we already know that. Here's the good news. If you understood chapter 12 and you understand mitosis, then you're probably going to understand meiosis pretty well, because meiosis is actually kind of basically mitosis times 2. And if you went and watched the crash course video, you'd definitely see what I mean. So I highly suggest go take a look at the crash course video if you haven't already. That always helps. I think he does a great job. And then I think I might have even put up Bozeman's science videos. He also does a great job. And of course, hopefully what I'm doing here is helpful as well. So let's take a look at this last chapter concept, 13.3. Like mitosis, meiosis is preceded by the replication of chromosomes. So just like in mitosis, there's like an S phase here where the chromosomes replicate and duplicate themselves. And then meiosis actually takes place in the two sets of cell divisions. So we have what we call meiosis 1 and meiosis 2. And the good news is they follow the exact same path. There is a prophase, metaphase, anaphase, telophase, or your textbook talks about prophase, prometaphase, metaphase, anaphase, and telophase. But basically there is a variation 1 and a variation 2, but they do follow a lot of the same trajectory and a lot of the same things happen. We're going to mention some of the details because particularly in the first stage, meiosis 1, there's some slight differences, some important things that go on. These two cell divisions, in the end, we're going to result with 4 daughter cells rather than 2 daughter cells in mitosis. And the big difference is that each of these daughter cells only has half as many chromosomes as the parent cell. So in mitosis, you ended up with 2 daughter cells that have the full set. In meiosis, you're going to end up with 4 daughter cells, each of them having half a set of chromosomes. And then there's some other details that make it important. We'll get into that as well. So we can talk about the stages of meiosis. So we kind of have like part 1 or act 1 and act 2. After the chromosomes duplicate, just like they would in mitosis, you have 2 divisions. So you have meiosis 1, which we can call the reductional division. This is where the homolog pairs line up or they pair up, and then they're going to end up separating. And so you're going to end up with 2 haploid daughter cells with replicated chromosomes. So after meiosis 1, you basically end up with 2 daughter cells that are haploid, but they have the replicated chromosomes inside. And then in meiosis 2, which we call equational division, those sister chromatids separate. So in the end, you're going to have 4 haploid daughter cells with unreplicated chromosomes. And again, what I find is saying all this starts to make it sound way more confusing than it is. So let's take a look at some illustrations, and I think it'll be a lot more clear. Okay, so we're going to use a model here. We're going to use a model that has a very small amount of chromosomes. So we only have 2 chromosomes, or 1 single pair of chromosomes. That makes it a lot easier to see. But just imagine this, times 23, basically, and you'd be talking about a human. So here we have a cell. Here are the chromosomes, the homologous pair of chromosomes in the diploid parent cell. So it's diploid because it has the full set, 2 copies. I'm sorry, 2 homologous chromosomes for this particular set. So let's say the blue one came from dad and the red one came from mom. Chromosomes are going to duplicate just like they did in mitosis. So here in interphase, in the S portion of interphase, you've got duplication of chromosomes. So now we have basically a double copy of the chromosome from dad and a double copy of the chromosome from mom. These are sister chromatids. They are duplicate copies and they are connected at the centromere. These 2 sets are what we would call the homologous chromosomes. So this one from mom, this one from dad, but they're homologous to each other. In meiosis 1, we have all of the stages that we're going to go through: prophase, metaphase, anaphase, telophase. We'll talk about that in just a moment. But the end result is these 2 sets of chromosomes, the 2 homologous pairs, are separated from each other. And so you end up with 1 cell that has one side of the homologous pair. This would be, say, the father side, and this would be the mother side. And they have the duplicate copy in there, so they have the sister chromatids. So this is kind of the result of meiosis 1, which is the first act of meiosis. Then in the second act, they undergo further division where the sister chromatids are pulled apart. And you're going to end up with 4 daughter cells. Each one of them is haploid. They only have half of the genetics of the parent cell. And notice by random choice, we'll see in just a moment, random segregation here, You have these two had the mother side, these two had the father side. So what you end up with is 4 daughter cells, each only having half the genetic information of the parent cell. So meiosis 1, meiosis 2, and again meiosis 2 undergoes the same prophase, metaphase, anaphase, telophase, just like mitosis did, but basically we're doing this twice, and we'll find out there's a little bit of special activity that goes on as well. So let's start with meiosis 1. Meiosis 1 is preceded by interphase. So we already talked about interphase, where the cell is growing in size and it's going through the G1, the S, and the G2. In the S, they duplicate their chromosomes to form those sister chromatids. The sister chromatids, remember, are genetically identical and they're joined together at that waist, that narrow area called the centromere. The centrosome, which forms the spindle fibers, replicates, forming 2 centrosomes, and those 2 centrosomes move toward the poles, and they start to spit out those spindle fibers that help line up the chromosomes. All of this is exactly like in mitosis. For a good overview, you want to go see the BioFlix video that's loaded up on the website for meiosis. It's a great overview and kind of gives you all the basically the visual details of the process that's going on. So once we've gone through interphase and the cells have replicated their chromosomes, we're ready to go through our first division. In meiosis 1, has the same 4 phases as we saw in mitosis. You have prophase, metaphase, anaphase, telophase, and then you have cytokinesis. So in your textbook, you can take a look on page, let me find the page for you. Looks like page 254 and 255. Great illustrations there. But you can kind of see what's going on. It's very similar to what happens in mitosis. You'll notice there's a little bit of funny stuff going on here where the homologous chromosomes are kind of overlapping each other. We'll discuss that in just a moment. But if you want to look at the basics, you kind of see where the chromosomes thicken to where they're visible. The centrosomes start to spit out the spindle fibers, the microtubule spindle fibers. You go on to metaphase, where everybody starts to line up in the center there. In anaphase, they're pulled toward the poles. And then in telophase and cytokinesis, you have this division and the reformation of the nucleus. All very similar. Now, we are going to find out there are some odd things going on in prophase 1, but we'll talk more about that in just a second. So here we are in prophase 1, and we're going to talk about what makes this process of meiosis unique and how does it result in differences from the daughter cells than you would get in mitosis. Prophase 1 typically takes up most of the time for meiosis. This is where the chromosomes begin to condense, and you see they're usually visible through the microscope. And then you have what's called synapsis, which is where the homologous chromosomes kind of pair up together, kind of aligned gene by gene. So you have the duplicated father chromosome and the duplicated mother chromosome, the homologous pair. They kind of sort of line up together side by side, and they're lined up in a way that aligns the genes on those chromosomes. If you want to see what's going to happen here, if you haven't seen the crash course video, go take a look at that. And this is prophase 1. This is where the action called crossover and recombination, homologous recombination occur. So again, look for that in the crash course video, and we'll talk about it here. In crossing over, what happens is the non-sister chromatids exchange DNA. So we'll show a picture in just a moment to make it more clear. But you have the 2 homologous pairs. You've got the father DNA strand and the mother DNA strand, and they are so closely together that they kind of overlap. And in areas where the chromosomes overlap, they actually swap some DNA. They actually swap segments or genes on those chromosomes. So they form what's called a tetrad, which is a group of the 4 chromatids. So remember, you have the 2 sister chromatids, and if you have 2 pairs, you have a 4 called a tetrad. And those tetrads have a couple of spots where they cross over each other, like they get intertwined. And this is called a chiasmata. I believe that's how you pronounce it, chiasmata. These are the X-shaped regions where they tend to cross over and sort of interconnect with one another. And this allows this swapping of DNA. We'll take a look at a picture and it'll show you some more. What's cool about this is called recombination. This allows for the creation of new, unique genetic coding on that particular chromosome. So I ran and grabbed an illustration off the internet just to kind of show you. So here we have the homologous chromosomes. So let's say the blue one came from dad and the kind of tannish one here came from mom. Notice that they have replicated in the S phase. They replicated. So here you have two copies. They are sister chromatids joined at the centromere, but these two are homologous pairs to each other. So they kind of line up together, and then they have this crossover effect that occurs. They basically become intertwined or intertangled, and there are these little X regions where they do overlap. This is called a chiasma or chiasmata. Not really sure how to pronounce that. I've seen it pronounced a couple different ways, but these crossover points are right here. And then what happens is they actually will swap pieces of genetics. So what you end up with at the end of this recombination is, take a look, you have differences. Now this chromosome right here, this one that's on the lower portion of what we call the mother, notice it's different from its sister chromatid now because it has a piece that came from the father chromosome. And then look at the two father sister chromatids. This top sister chromatid is now different from the lower sister chromatid because it has a swapped portion that came from the mother's chromosome. So this is all happening in prophase 1, where you have these chromosomes, they get together as homologous pairs, and then you have this crossing over, and then you have what's called recombination, where they actually swap pieces of their genetic code, and this ends up creating brand new possibilities. So this is all prophase 1. This is where the real action happens in meiosis. All right, moving on to metaphase 1. If you'll remember, metaphase is where everybody kind of lines up along that center plate in the middle. So we have the tetrads, which are the homologous pairs that have been duplicated. They line up at the metaphase plate, which is in the center of the cell, which one chromosome facing each of the poles. We have this microtubule structure that comes through and attaches to the kinetochores in the center of the chromosomes and kind of pulls them one way, left or right or up or down, and kind of centers them in the middle. So the microtubules are connected to the center portion called the kinetochore, and that pulling back and forth lines them up in the very center called the metaphase plate. Then we move to anaphase 1. We know in anaphase the chromosomes are pulled apart. So here what we're pulling apart is not the sister chromatids, we're basically separating the homologous pairs. So we're pulling the mother chromosomes one way and the father chromosomes the other. So we're not breaking that connection between the sister chromatids, we're simply separating the homologous pairs, moving them toward each pole. Then we have the formation, again, sort of a nucleus and the splitting apart of that cell, and that moves us into the telophase and cytokinesis. So in the beginning of telophase 1, each half of the cell has a haploid set of chromosomes, but they are a duplicated haploid set. Each chromosome still consists of two sister chromatids. And then we have the cytokinesis, where the cytoplasm material pinches together and forms two haploid daughter cells. Let's take a look at a picture. Always better to look at a diagram. So we already know in animal cells, the same way as in mitosis, a cleavage furrow forms, and in plant cells, a cell plate forms. It's important to know that no chromosome replication occurs between the end of meiosis 1 and the beginning of meiosis 2. So we already replicated the chromosomes in the S phase of interphase. We're not going to replicate again. So once we get started with meiosis 2, there won't be replication of chromosomes prior to meiosis 2 like there was prior to meiosis 1, because remember, we're going to split all this in half and create 4 daughter haploid cells. So one important thing to remember is, unlike in meiosis 1 and mitosis, where you have chromosome replication prior to the beginning of it, you will not have chromosome replication prior to the beginning of meiosis 2. So going back to our picture to look at meiosis 1, prophase, metaphase, anaphase, telophase, all with a 1 after it. Really the only difference here, or probably the most important things to remember, is the point at which the homologous pairs line up together and you get this crossing over, or the chiasmata, and you get this swapping of genetic information. Then the homologous pairs line up along the metaphase plate, and here at anaphase, we're not separating the sister chromatids, we're separating the homologous pairs into 2 daughter cells. Now we're ready to go to stage 2, or the second act of meiosis, called meiosis 2. All we do is throw a 2 on the end, so we have prophase, metaphase, anaphase, telophase. Meiosis II is very similar to mitosis. You don't even have the special activity that went on in prophase 1. So here's an overview: prophase, metaphase, anaphase, telophase. This is almost exactly like you would see in mitosis, where you have the chromosomes begin to condense, you have the centrosomes creating the microtubule structures, they line up on the plate in metaphase, the sister chromatids are now pulled apart in anaphase, and then you have telophase or telophase and cytokinesis at the end. Prophase 2, spindle apparatus forms. In late prophase 2, the chromosomes begin to move toward the center. They have condensed to where they're visible. Metaphase 2, the sister chromatids are arranged at that metaphase plate in the middle. And because of that crossing over that we had in meiosis 1 and prophase 1, the two sister chromatids, remember, are no longer genetically identical. So unlike in mitosis, the two sister chromatids now are slightly different from each other. So going back to our picture here, so see how this sister chromatid at the very bottom no longer is an exact match of this sister chromatid that it's connected to. And this sister chromatid right here is no longer an exact match of the sister chromatid it's connected to. So that's one difference in meiosis. So once we're all lined up in that center line along the metaphase plate, we pull apart. In anaphase 2, the sister chromatids are separated and they are pulled toward the opposite poles. And then we have telophase and cytokinesis, where we have the pinching off of the cytoplasm. The chromosomes begin to unravel and take their usual form, and we have the formation of the nuclear envelope again, and we are back to where we kind of started with. But now we have 4 daughter cells that are genetically unique, and these 4 daughter cells are also haploid. So the end result of meiosis is 4 daughter cells. Each of them have a half set of chromosomes, and each of them is genetically different from each other and genetically different from the parent cell. If we're talking about males, these 4 daughter cells all become functional sperm cells, and they could implant with an egg and begin the whole process of forming a zygote and the whole reproductive process. If we're talking about females, it's a little bit different. In females, these 4 daughter cells, through the whole process of meiosis, one of these daughter cells tended to get more of the cytoplasm, more of the organelles and things like that. So one of these 4 daughter cells tends to be larger and more well composed than the rest. That one will become the egg. The other 3 form what's called polar bodies, and it depends on the species, but for the most part, these polar bodies are not part of the reproductive process. So of the 4 daughter cells, in males, all 4 become active sperm cells. In females, only one of those 4 daughter cells becomes an egg. The other 3 form polar bodies, and what happens to them is dependent upon the species. So let's kind of do a quick comparison of these 2 types of cellular reproduction, mitosis and meiosis. Mitosis conserves the number of chromosomes. We are producing genetic clones, identical copies to the parent cell. Meiosis reduces the number of chromosomes in half from diploid to haploid and produces cells that are genetically distinct from each other and genetically different from the parent cell as well. And a picture, of course, is always good. Whoops, kind of hit the mouse. Let me get you back to where we need to be. An illustration is always a good thing. So here's a great illustration taken from your textbook that kind of gives you the simplified version, of course. Breakdown of mitosis and meiosis. Mitosis, we end up with 2 daughter cells that have the full complement of genetics. In meiosis, we end up with 4 daughter cells that are genetically distinct, and they have a half set of the genetics. Here we are in words, just a quick summary. So this is another good slide if you needed a good comparison. Oftentimes a Venn diagram or comparison like this helps you understand. So this talks about particular properties, and then mitosis compared to meiosis, what you get from the 2 different processes. So although mitosis and meiosis share a lot in common, they're very similar, there are definitely 3 events that are unique to meiosis and very important, and these all occur in meiosis 1. So primarily in prophase 1, as a matter of fact, but all in meiosis 1. So we have synapses, which is where the homologous chromosomes kind of get together and get side by side, and they become intertwined. They cross over. This is in prophase 1. So the homologous chromosomes physically connect and exchange genetic information. This is recombination. So now the sister chromatids are not exactly duplicates of each other. And then at the metaphase plate, again, this is in meiosis 1, at the metaphase plate, we have tetrads, the homologous pairs that have been duplicated. And when we separate, when we pull those apart, we're not pulling apart the sister chromatids, we're separating those homologous pairs. And then in anaphase 1, oh yeah, that's in anaphase 1. In anaphase 1, when we pull those apart, we're separating those homologous pairs, not the sister chromatids. So probably the most important event is this crossing over and recombination, because that's what's going to allow for these genetic differences, these unique daughter cells, which is going to add to the genetic variety of the species. The book does go into some detail about how these separations occur. I probably wouldn't ask a lot of questions on a quiz or a test about this, but it's good to know that the sister chromatids stay together through a process called cohesion, and there are certain protein complexes called cohesins that basically hold them together. This is kind of like the glue that holds the sister chromatids together. And in mitosis, these cohesins, these glues, are broken or cleaved in metaphase, by the end of metaphase. So the connection that holds the sister chromatids together is broken, and that allows them to be pulled apart at the end of metaphase and then into anaphase. But in meiosis, these particular proteins are not broken, so the sister chromatids stay together, but the cohesive proteins that hold the homologous pairs together are broken. And that allows the homologous pairs to be separated in anaphase. And then when we get to the second stage, anaphase 2, that's when those cohesive proteins are broken between the sister chromatids and allow them to separate. So basically this is just the molecular basis for how the chromosomes behave. And there is still a lot to be learned about this. So this is still an area of intense scientific study. Exactly how does this crossover and recombination occur, and what controls all of that? We know a lot about it, but there's still a lot to be learned. So this is still an area, genetics in particular, is an area that is still very much in study, and genetic scientists are very much in demand. Let's wrap it up with concept 13.4. How does all of this relate to the species and the evolution of species? It's obviously a very important part. Concept 13.4 says genetic variation from meiosis produced in these sexual life cycles contributes to evolution. Now, there are a couple different ways you can get genetic variation. One of those is mutation. So a mutation is a change in the organism's DNA. That's the original source of genetic diversity. But then, through meiosis, you have this swapping of genetic information that allows greater diversity to occur. And so mutations create different versions of genes called alleles. And then through meiosis, you have a reshuffling of these alleles in sexual reproduction. So the hallmark of sexual reproduction is the ability to produce genetic variation. Also, keep in mind when we talk about mutations, most people, when you say the word mutation, they think of something negative or bad. But in biology, in science, the word mutation simply means a change in the genetic code. And sometimes this can be negative. Sometimes this can be so negative that it results in the death of the creature or the embryo not fully forming or something like that. So a mutation, a genetic mutation, can be negative, but a genetic mutation can also be positive, or in many cases, it can simply be inconsequential. In other words, it has no particular negative or positive effect whatsoever. So mutation simply means a change in the organism's DNA, not necessarily negative. It can be positive, or in many cases have really no effect at all. So mutations are the original source of genetic diversity, and then through meiosis and sexual reproduction, we have a shuffling of all these genetic cards that allows for a whole bunch of genetic variation and diversity. Through sexual reproduction, we have kind of three main things that allow you and your siblings to look different from each other and not be exact clones. And for if you have a litter of 6 puppies, how they may not all 6 look exactly alike or something like that. We have what's called independent assortment of chromosomes. We have what's called crossing over, which we've already talked about, and random fertilization. So quickly, we're going to look at those three. And these three kind of help explain this genetic variation that you see in sexual reproduction. First one's called independent assortment, and that means that what one chromosome does doesn't affect what another chromosome does. So remember, in metaphase 1, we have all of the homologous pairs begin to line up along the center line, along the center plate there. Well, they do so completely independent of each other. They don't know what the other chromosome is doing, and it doesn't affect their behavior. So you could have the parent, the mother chromosomes, line up toward one pole, and the father chromosomes line up toward another pole. And then the next chromosome, it could be opposite. You could have the father chromosome line up this way and the mother this way, and the next one could be opposite or it could be the same. So what each chromosome pair, homologous pair, does along that center line is completely independent of every other pair. Well, that means there's a 50-50 chance that the mother chromosome will be on this side and the father chromosome will be on this side. That chance is called independent assortment, and that plays a part in the variety that you might see. Since we're talking about chance, let's get into a little bit of mathematics. Based on that independent assortment, the number of combinations that's possible when the chromosomes assort independently, randomly like that, is 2n, where n is the haploid number. So mathematically, for humans, n is 23. We have 23 pairs of chromosomes. And so that means there are more than 8 million, 2 to the 23, 8 million possible combinations of chromosomes. So the formula is 2 to the n, where n is the haploid number. And that's because each chromosome can arrange itself completely independently of all the others. So you have 23 pair of chromosomes independently arranging themselves. That's a lot of possible arrangements and a lot of possible combinations. And mathematically, it is 2 to whatever the haploid number is, 23. So 2 to the 23, that's more than 8 million possible combinations of chromosomes. And that is just taking into account independent assortment at that center line. That doesn't even take into account all the possibilities that arise from crossing over and recombination. So that one part accounts for 8 million, more than 8 million possibilities. Here's a diagram that talks about that independent assortment. So in this cell, we have only 2 sets of chromosomes. So a very small number as opposed to 23 pair that we have. We have 2 pair here. And again, the chromosomes have duplicated because they've already gone through the S phase. So here are the homologous pairs, one from dad, one from mom. And then here's a homologous pair, one from dad, one from mom. Now they could line up this way, or the other possibility is they could line up this way. So you have 2 possibilities. Now you could say, well, what about if the red ones line up on the left and the blue ones line up on the right? Well, really, that's just a variation of this possibility. So we have 2 possibilities. Each one is equally probable. They don't interact with each other, so each one of these could happen at metaphase 1. So here at metaphase 2, then again, you have this possibility here, or this possibility, or these 2 possibilities here. So what you end up with is all of these possible combinations. Combination 1, 2, 3, or 4. So because of independent assortment, you end up with all these different possibilities. And that's simply based on how the chromosomes line up in the center and all the possible combinations of how they might line up. That's called independent assortment. Now, let's get to that crossing over part. Remember, the crossing over is that recombination where the homologous pairs line up and they actually swap portions of their DNA. This starts to happen early in prophase 1 as they begin to line up side by side. In the crossing over, the homologous portions of the 2 sister chromatids trade places. So where you have that chiasmata, that crossover point, you have chunks of DNA basically switching from one chromosome to another. And this contributes to genetic variation because now you have chromosomes, sister chromatids, that are unique and different from the way that they used to be. Let's take a look at a picture. So here we have 2 homologs that are lined up very closely together, 2 homologous pairs. You have some points at which they cross over. You can see them connected right there. They swap portions of the DNA, the genetic code. So now you notice this one has a bit of the red, this one has a bit of the blue. Now you notice that the two sister chromatids no longer match. They're no longer exact copies. Then we go through the whole process of meiosis, separating those out, to where you end up with daughter cells that are different from each other. This daughter cell is different from this one, from this one, from this one. In this case, you can see where they have recombinant chromosomes, where they have taken pieces from the other chromosomes. So this ends up with a lot of variety, this process. Last but not least, the last thing that allows for such a huge variety is what we call random fertilization. And random fertilization means that any egg can fuse with any sperm. So any sperm can fuse with any ovum, which is an unfertilized egg. And when two animals reproduce, there's no particular way to know which sperm is going to combine with the available egg. And there are hundreds, thousands of potential combinations. So there are hundreds and thousands of potential sperm cells. You have no idea which one of those is going to form the zygote with the particular egg that we're discussing. So that's random fertilization. So you take all of those factors, combine them, and you end up with an enormous possibility of variety. So think about the numbers involved here. When you create a zygote, you have the fusion of two gametes. Each of those gametes had about 8.4 million possible chromosome combinations from independent assortment, and they produce a zygote that has about 70 trillion possible diploid combinations. And then you throw in crossing over, the whole recombination process, that adds even more possibilities. So at that point, the number of genetic variations is astronomical. It would take a lot of zeros, let's put it that way. So your parents could have trillions of children, and never would two of them come out to be exactly the same. So basically what we can say is that each zygote has a unique genetic identity. While you share a lot of genetic information with your mom and dad, you share huge chunks of genetic information with your siblings, your brothers and sisters, each one of you is unique and separate when you get down to all the little details of your DNA. So when someone says, man, you're 1 in a million, you're unique, well, it's more than that. You are 1 in trillions. So definitely your code is unique, and while you share a lot of common characteristics and traits, you are not a clone of mom or dad or brother or sister. Now, unless you're an identical twin, perhaps we'll talk about that later, but identical twins are basic genetic clones of each other because they actually began as one single fertilized egg that divided and created two embryos. But that's a separate story. So as far as we're concerned in sexual reproduction, it produces an enormous amount of variety, and you yourself are 1 in trillions of possibilities. Here's a great little animation that talks about these three things that allow for genetic variation. You have independent assortment, you have crossing over recombination, and then you have random fertilization. So you might want to right click and hit play on this. You should be able to see this animation. The guy's voice is kind of boring, but the animation and the pictures really help you understand the material. Last but not least, let's talk about the evolutionary significance of all this genetic variation within the population. Of course, throughout our textbook and throughout our study biology, we talk about evolution quite a bit. And this genetic variation that we're discussing in this chapter, in this sexual process of meiosis, has a lot to do with the possibilities of evolution and changes over time. And Charles Darwin, and indeed other scientists at the time, recognized that populations evolve through differential reproductive success of its members. So you have some members of a species that are better at surviving and reproducing than others. And on average, the ones that are best suited for their local environment, for whatever reason, perhaps they're better camouflaged, or they have a shape of beak that makes it easier to get food, or they are a little bit faster, or whatever physical variation that they have, the ones that are best suited to their local environment tend to leave the most offspring. And that's obvious because they tend to live the longest and they tend to have the most reproductive success. So they tend to pass on their genetics over time that causes the species to evolve and change in response to environmental conditions. So natural selection results in the accumulation of these genetic variations that are favored by the environment. So if you have, say, frogs that live in a muddy area and the mud is kind of a brown color, well, you're going to have natural variation. Some of the frogs are going to tend to be sort of brown that matches the mud. Some of them may be sort of green. Well, the ones that tend to do best are the ones that escape predators and have other physical variations that help them survive. So over time, you might expect that the species would tend toward more brown offspring, and that tends to be the case. That's natural selection at work. Sexual reproduction is what allows all the genetic variation that natural selection works off of, with a combination of mutations and then this reshuffling of genetic material through sexual reproduction. These are the ingredients, basically, that then natural selection works with to produce species. Now, it may be important to ask, do all animals produce sexually? Because there would seem to be obvious benefits, and there are. The genetic diversity is the greatest benefit, but it's also highly energy intensive. It's difficult to look for a mate and find a mate and keep a mate and raise a child. So there are downsides to sexual reproduction. And indeed, there are some creatures that reproduce asexually. And this is a good example. This is called a deltoid rotifer. It's a microscopic creature, so it's not like something you're going to go to the zoo and see. But it reproduces only asexually. And you would think, okay, well, it's doing nothing but making clones of itself, which has some disadvantages. It can't really evolve much in response to changes in the environment. But if it's already well suited to its environment, that might be a good thing just to produce clones. And so there are some examples where asexual reproduction is what goes on in some particular animals, like in this case of this particular creature. But you're going to find out later that even in this creature that reproduces asexually, there are methods of modifying the genetics. There's something called horizontal gene transfer, and there are other ways that this creature uses to diversify its genetics. So sexual reproduction is by far the most common method in the world of living creatures that we see for creating genetic diversity, but it's not the only method. We'll talk about some later on, I think in chapters 26 and some of the later chapters. Finally, let's look ahead to our next chapter. So next chapter is all about genetics, or a little bit about genetics. And even though Charles Darwin and some of the other scientists of his day, they understood or they realized that variation in heritability is what makes evolution possible, but they couldn't really explain why offspring are oftentimes a lot like their parents, but not exact copies. Darwin and the other scientists of his particular time, they didn't know or understand genetics. They had no knowledge of genes and chromosomes and things like that. It just wasn't something that scientists knew anything about at that time. And there is a little bit of irony, because the next scientist that we're going to look at is one of my favorites, Gregor Mendel. He was a monk, an Augustinian friar, and he actually lived at the same time Darwin did. And he actually published some papers about the theory of inheritance. And if you've ever heard the story of Gregor Mendel, you know he was working with pea plants in his garden. And his findings helped explain this genetic variation. He's considered the father of genetics. But the irony is that his published papers and reports didn't really get to the right people that were studying this information. And so it didn't really cause much of a stir. And actually, Darwin, even if I'm not even sure if perhaps he even knew of Mendel's work or did not, but at any point, the connection wasn't made. And as a matter of fact, the connection wasn't made until about 1900, the early 1900s, and this was after the death of Darwin, who died in the late 1800s, 1882, and Mendel in 1884. So while Darwin understood the process of natural selection and evolution, he couldn't really explain what was the fundamental cause of this. He didn't understand genetics. Gregor Mendel did begin to discover a little bit about genetics, but he died before his findings were actually realized and came to be worked on until we are to the point now where we understand so much about genetics. And the cool thing is, I think it would be awesome if we had a time machine and both of these guys, Charles Darwin and Gregor Mendel, could see what we know today. I think they would be astounded at how much information we have and what we have today that helps verify what they were doing way back in the 1800s. So that's it. We're going to stop right there. We'll stop with the little pea plants and Gregor Mendel. We'll talk more about Mendel and genetics coming up in Chapter 14. Have a great day or weekend or whatever it happens to be for you, and I'll catch you for the next chapter. Bye-bye.